Welcome to The Explainer. Today we are doing a complete slide-by-slide summary of a truly fascinating internal scientific presentation called Targeted Biological Blueprints. We're talking about what could be the next massive paradigm shift in regenerative medicine. We are moving away from generalized one-size-fits-all treatments and zooming in on something incredibly precise, using targeted biological blueprints for true, localized organ repair. Okay, let's dive right into this. Here is how we're going to break it down. First, we'll cover the new therapy paradigm. Second, we've got a brain case study focusing on OPCs. Third, a kidney case study on podocytes. Fourth, we'll synthesize that into the instruction packet model. And finally, we'll look at the future protocol and roadmap. Starting with Section 1, the new therapy paradigm, and the core problem with current MSCs. For years, the medical approach has been almost entirely systemic. Pumping generalized stem cells into the body and just kind of hoping they find the damage. But this blueprint argues a bold new thesis. True organ repair requires highly specific signaling packets. To actually fix the damage, we have to transition toward localized, targeted secretome delivery using highly specialized cells. Think of systemic stem cells simply as an ignition switch. They turn the engine on, they provide a temporary pulse of immune relief and improved microcirculation. But they don't stick around to do the actual building. For that physical rebuilding, we desperately need specific voice messages. These are targeted payloads delivered directly to the damaged tissue. Let's really contrast these two. Systemic therapy is basically a hit-and-run payload. Research shows these MSC cells have an almost 0% survival rate in autologous environments, meaning inside the patient's own body. They last, maybe 24 hours? Localized secretome therapy, however, utilizes immortalized cells to deliver robust, continuous, highly specific rebuilding instructions exactly where they are needed most. Moving on to Section 2, Case Study, Brain OPCs. Let's see how this localized repair theory applies to neurological degeneration. Let's track how this builds. We start out with a raw, generalized neural stem cell, which moves to a neural progenitor, then into an oligodendrocyte precursor cell, or OPC, and finally it becomes a mature oligodendrocyte. The ultimate goal here? Creating the myelin sheath. That's the protective insulation around your neurons that, unfortunately, breaks down in debilitating diseases like multiple sclerosis. So why isolate the OPC specifically? Well, it's the ultimate Goldilocks cell. A fully generalized stem cell doesn't really know what to do yet, and a fully mature cell has already stopped building. The OPC sits perfectly in the middle, actively secreting the exact biological blueprints we need to physically rebuild those neural pathways. But we are fighting a major biological ticking clock here. These cells are robust and viable for only a tiny window, between about 2 weeks and 3 months post-birth. After that peak activity window, biological signals, specifically the PDGFA trigger, actually force the cell to mature, completely slamming that viability window shut. To capture that magic, scientists have to use a manufacturing protocol with an incredibly strict 12 to 24-hour critical window. Once the tissue is plated, the clock starts. To stop them from maturing, scientists introduce SV40 and hTERT viral immortalization. This is essentially a biological pause button. It freezes the cell right at its maximum therapeutic potential, letting it live and secrete those repair instructions indefinitely. Shifting gears now to Section 3, Case Study, Kidney Podocytes. We're diving into one of the body's most mechanically complex filtration systems. During the internal discussions behind this blueprint, one of the scientists actually said, "I've always felt the kidney is truly incredible. Its structure is much more complex than the heart." And as we're about to see, this sheer engineering marvel makes it uniquely desperate for targeted instruction packets. Let's trace the macro architecture down into the nephron, landing squarely inside Bowman's capsule. That is where we find the glomerulus tuft. To put this into perspective, in a human, this entire tuft is a mere 0.2 millimeters across, yet it powerfully manages our massive systemic filtration. Zooming in even further reveals the ultimate gatekeeper, the tri-layer filtration barrier. Layer 3 is especially wild. It's this interlocking, puzzle-like structure made of podocyte foot processes. They actively wrap themselves right around the capillaries to determine exactly what stays in your bloodstream and what gets filtered out. This whole system relies on opposing physics. You have hydrostatic pressure pushing fluids out and oncotic pressure pulling them back in. This creates a completely uncompromising size threshold. Essential small molecules, like insulin, pass right through, but critical larger proteins, like albumin, blocked and safely kept in the blood. Now, here's a massive discovery from 2023 that changes everything we thought we knew. Podocytes aren't just simple mechanical filters. They actually act as antigen-presenting cells, meaning they serve as highly localized immune modulators. Controlling this immune response right at the podocyte level is absolutely key to halting chronic kidney disease. And this brilliantly illustrates the tragic biological flaw we're dealing with. Mature podocytes are terminally differentiated. They cannot organically regenerate. If they get damaged by systemic issues like high blood sugar, they just permanently collapse. That causes proteinuria, where vital proteins leak into the urine, inevitably leading to dialysis. Because they can't rebuild themselves, we literally have to deliver therapeutic secretomes directly to them to save the filter. Which brings us perfectly to Section 4, the instruction packet model. Let's synthesize all of this. Recalling our earlier analogy, the model requires two distinct inputs. Input 1 is the systemic prep. MSCs are the ignition key, sending a pulse wave to prepare the degenerating environment. Input 2 is the local blueprint. Exosomes derived from immortalized organ-specific cells are injected directly into that degenerating organ, delivering the exact instruction packet required for physical rebuilding. Just to quickly walk through the comparison matrix, systemic MSCs are our high-expansion, hit-and-run regulators given via IV. In stark contrast, the OPCs in podocytes are incredibly specific. They require localized payload delivery and viral arrest to actually create stable, therapeutic cell lines capable of directing repair. So the ultimate clinical conclusion here is a two-pronged treatment protocol. Phase 1 deploys systemic MSCs intravenously to turn the engine on and dampen generalized chronic inflammation. Phase 2 follows up immediately with a targeted, localized injection of immortalized cell secretomes straight into the target, like the spinal cord or kidney cortex, to direct the actual physical repair. Finally, Section 5, Future Protocol and Roadmap, the immediate strategic next steps. So, the crucial point is, turning this blueprint into a clinical reality relies on 3 immediate milestones. First, perfecting that strict 12 to 24-hour window to get stable OPCs. Second, successfully immortalizing human podocytes. And third, mastering payload delivery design. We need specialized encapsulation so these localized packets survive until they fully penetrate their specific targets. This whole presentation represents a massive paradigm shift in how we think about healing the human body. It forces us to ask, will localized instruction packets finally conquer chronic organ failure where systemic stem cells have continually fallen short? Delivering the precise blueprint to the exact right location certainly seems to be the future of regenerative medicine. Thank you for joining me for this explainer, and keep questioning how the building blocks of tomorrow are being engineered today.